So welcome, Subbu. Thank you so much to accept this invitation. I believe our professors, our students will enjoy your presentation. I enjoy your class as a student, and I learned a lot. And with your experience in the industry and with academia, I think it makes this presentation quite unique. So we talk, of course, a lot about the patches drug delivery in my class and the stents and a lot of things that, of course, you have been doing. So I believe it will be a great webinar for us. Thank you so much, Subbu, to accept again this invitation, and you can start anytime.
Okay, thank you, Frank. It's my pleasure really to be able to do this webinar. It's of course 7 a.m. here, so I may not sound awake all the time, but I will try to. And what I'm going to do is now share my screen and talk about the evolution of cardiovascular implant biomaterials. I hope everybody can see this.
Yes, we can see well, Subbu.
Okay. All right, so basically I have subtitled my talk, Fixing Broken Hearts, the Evolution of Cardiovascular Implant Materials. And I'll talk about sort of three broad areas of cardiovascular disease or conditions that are addressed by new cardiovascular biomaterials. But hand in hand is the evolution of cardiovascular medical devices. So a lot of the innovation in medical devices is driven by the use of new materials, the clever use of new materials, and vice versa. So many of the new devices require input from material scientists. So I used to be at Nanyang Technological University, where Frank also got his PhD. But recently I've moved over to the National University of Singapore, where I'm handling their intellectual property portfolio, as well as my own research. But before I actually start talking about cardiovascular implant materials, I want to put the evolution of these materials in the overall context of biomaterials research, biomaterials evolution. So if you start to look at how biomaterials in general have evolved over the years, and I'm old enough to have followed most of the developments here. So we started out with bio-inert and biostable materials. Examples are hip joint materials or knee joint replacement materials, or the intraocular lens that are used for cataract patients, metal stents, and so on. These were all materials that were supposed to be inert to the body and stable over time. They did not degrade or do anything with the surrounding tissue. From here, we moved on to what we call biostable and bioactive implants, which include drug-eluting stents and orthopedics that are coated with a layer of, for example, hydroxyapatite that allows them to be integrated with the tissue. In the case of the drug-eluting stents, of course, they were supposed to be bioactive against reclosure of the stented segment of the arteries. Then we moved on to biodegradable and biointegratable devices, such as tissue engineering scaffolds, stents and occluders, which I will talk about later on today in more detail. And then the current situation is that everybody is looking at biodegradable, but also bioactive. So these are scaffolds, for example, that allow the guided differentiation of stem cells after being implanted, just as a material, not with any biochemical cues. So the material itself is, for example, conductive and is able to guide the differentiation of neuronal cells, stem cells, and so on. So this is the current area of research. And of course, in parallel, there have been a lot of work in biomaterials for drug delivery. I will not be talking about any of that today. And the role that polymers play, which is my area of research, is highlighted here. So polymers play an interesting role in tissue engineering scaffolds, in the biodegradable bioactive, and so on. So I'll cover a lot of this involvement of polymers in devices in this talk. But right now, the other interesting trend in the evolution of biomaterials is the use of biomimetic materials. These are things that occur naturally, but are produced in the lab in larger quantities using synthetic biology approaches. And they can be applied across the entire set or entire classes of biomaterials. And that's a very exciting area, which will naturally produce biocompatible materials, which are also processed in an environmentally friendly fashion, because most of the natural synthesis of these materials occurs in aqueous media. So that is environmentally very friendly. So that's a very exciting area, but unfortunately, I won't have time to go into that in this talk. So in our group, we are interested in polymeric biomaterials for various applications. This is just a slide to show that we are working on cardiovascular implants or therapeutic implants, ocular implants, as well as nanomedicine for ocular drug delivery and so on. And tissue engineering, we are working on cellular delivery systems for various kinds of tissue engineering applications. I do want to put out a disclaimer with regard to cardiovascular materials, because I did spin off a company called Amaranth Medical, which is headquartered in California, with a branch here in Singapore Science Park. So I do want to talk disclaim my association with a company that produces cardiovascular stents. So let me now turn to the applications. The first application I want to talk about is really addressing heart attacks or blockages in the coronary arteries that lead to heart attacks. This is my first example of a broken heart. And in this case, I'll talk about the evolution of materials used not just for stenting, but also for balloon angioplasty and so on. The second condition that I want to talk about is the case of occlusion, or blocking an axis that exists currently abnormally in the heart. So we call them holes in the heart, which should not be there, but which need to be occluded for various reasons. So the first example is of stents, which open up blocked arteries. The second example is of occluders that will block openings within the heart that should not be there. And the third thing I want to talk about are what are called embolic devices, which also block certain areas of the cardiovascular system. For example, to prevent aneurysms, you block off the access to the aneurysm of blood by putting in a plug right next to the aneurysm site. So I'll talk about these three examples of cardiovascular devices and the materials that play a role in these. So most of you are probably familiar now with Frank educating you about the problem of a blocked coronary artery, which leads to a heart attack or myocardial infarction. And that causes muscle damage downstream of the blocked artery. So the idea is to identify this condition as soon as possible, rush the patient to the hospital, and then open up the artery, the blocked artery in the heart. And traditionally, it was done by using what is called coronary artery bypass surgery, where an artery from a healthy vein, sorry, not an artery, from the leg or from the mammalian artery is excised, is cut off by the surgeon, and then that is used to bypass the blocked artery. Okay, and that allows for flowing of the blood to areas below the blocked artery, so that tissue function can be restored and tissue death minimized. So the problem with having the blocked artery is that the muscular tissue, especially, dies away with lack of oxygen and cannot be regenerated. So it's one of those tissues that the body does not regenerate naturally. So it has to be replaced with tissue engineering. But the idea is to arrest the problem very early on so that very little tissue is lost. But the bypass surgery procedure is very involved under general anesthesia. It is an inpatient surgical procedure, very costly. It takes at least a week to several days for recovery, and normal activity of the patient can only resume after many, many, many months. So very involved procedure, which was not very popular with the patients. So the first revolution in cardiovascular devices came with the invention of the balloon catheter. And this is what is called the angioplasty procedure, where a catheter is inserted into the blocked artery, and it's guided by X-ray fluoroscopy to the site of the blockage, the plaque, then expanded with balloon pressure, and that pushes the plaque to the sides of the artery. The balloon is then withdrawn. This opens up the artery, is then withdrawn, and then the patient goes away after a few hours in the outpatient clinic. So the key to this procedure, of course, is the fact that you can insert this balloon catheter, which is unexpanded, through an artery in the arm or in the legs. Okay. And that is minimally invasive. So there is no opening up of the heart. There is no cutting through the ribs to do open heart surgery. It's simply a catheter that is inserted into the cardiovascular system, guided, as I said before, by X-ray fluoroscopy over to the blocked site. And then the artery is unblocked. Okay. That is the procedure. And the inventor of this procedure is Dr. Charles Dotter, who won the Nobel Prize in 1978, is widely considered the father of interventional cardiology. So the term now goes away from cardiac surgery to interventional cardiology. And like I said, this is a separate department in many hospitals these days from cardiac surgery. And this gentleman should be given a lot of credit for inventing this procedure and opening up the whole area of minimally invasive surgical intervention. So cardiac surgeons no longer do many of these procedures. So going on with the story of the evolution of cardiovascular devices, what happened after the balloon angioplasty procedure was approved in the 1970s is that it was found that for those patients that were subjected to balloon angioplasty, there was a chance that the same segmented area, the same area that was blocked before could be re-blocked. So this is called re-stenosis or re-blockage. So balloon angioplasty solves the problem for 50% of the patients or thereabouts, but for many of them, there needs to be a repeat procedure to reopen the artery in the same place. And that becomes a little bit of a problem. So this restenosis led to the discovery of stenting in conjunction with balloon angioplasty. So stenting simply involves the use of a coronary stent, a metallic stent, that is crimped onto the outside surface of the balloon catheter. And then along with the balloon, it's expanded in place. But when the balloon retracts, the stent is still kept in place in the body. So it's a permanent stent that keeps the artery open for much longer than just the balloon angioplasty procedure. Okay, so that was approved in the 1980s. Sorry, in the 1990s. And then the problem of restenosis was reduced. So with the introduction of the stent, the problem of restenosis went from about 50% to about 30% or 25%. And the reblockage is actually a result of the stenting or balloon angioplasty procedure itself, because when the stent is expanded and the metallic stent pushes against the blood vessel tissue, it actually causes trauma to some of the tissue. And in response, the body produces more of that kind of cell, smooth muscle cells in this case, and that causes reblockage, again, at the same site as the stented segment. Okay, so this restenosis is not completely eliminated by the use of stenting, because the stent material itself causes some trauma to the surrounding tissue. This was then solved by the use of drug-eluting stents, where the surface of the stent is now coated with a drug-containing polymer, which then touches the tissue, slowly releases the drug into the tissue, and that sort of kills or arrests the proliferation of the smooth muscle cells. So that brought the restenosis rates down to about 10% or less in many cases. So this is sort of a summary slide that tells you the evolution of these procedures, starting with balloon angioplasties in the 1970s, bare metal stents approved in 1990 by the FDA, and then the drug-eluting stent was approved in 2003. And then we'll talk about the future of stenting in the 2003 and onwards. Okay, so that was the status in 2000, when we also first became aware of the problem of restenosis. So this slide sort of summarizes what I've been talking about in terms of the issues with bare metal stents, as they are called, no drugs, and the drug-eluting stents. So bare metal stents still have high restenosis rates, 20 to 30%. The material is permanent. Usually it's stainless steel or cobalt chromium steel, and it provokes foreign body response, can induce clotting. It can also interfere with some of the diagnostic procedures of the same patients for other conditions. So if they want to undergo MRI imaging, for example, for some other condition, the stents interfere with the diagnostic procedure with the MRI. So drug-eluting stents delays healing. Although it reduces restenosis rates, it delays the healing, meaning that most of the stents that are employed in the body must be covered eventually with the body's own endothelial cells. This is the process of healing. If that is not done, the patients have to be on anti-clotting drugs for the rest of their lives, which is not a desirable situation. So the introduction of DES reduced restenosis rates, but it also delayed the endothelialization of these stents. And therefore, it became necessary to continue with this anti-clotting therapy for much longer than with the bare metal stents. And it was also noticed that there was some late-stage thrombosis or blood clotting because of the presence of the permanent stent. And this has been documented in many, many publications. So just to summarize, there are issues with just simply the fully stable biostable stents, as well as with drug-eluting stents. So people started looking at biodegradable stents as a solution to these problems. So if you can design a stent to open the artery, be mechanically stable for a few months, several months, and then if it can disappear, then the problems with late stage thrombosis as well as delayed healing will go away. And also, you might wonder why stents should be made to disappear, because they are keeping the arteries open. It turns out that the permanent stents are really not needed in the body after 6 months. So following the stenting procedure, Patients have been evaluated for whether or not reclosure occurs. And like I said, if in the first 6 months, there is some reclosure, but if it doesn't happen for those patients after 6 months, there is no problem at all. So the stents are actually not needed after 6 months. Now, a number of studies have shown this. So it's actually redundant to keep the stents in place after 6 months. So then the idea of a biodegradable stents that disappears after 6 to 9 months came up in the 2000s, so to speak. What is required of the material is that it should be mechanically strong for about 9 to 12 months and then start to disappear. And then it should also have the provision for releasing drugs. But most importantly, it must be capable of being inserted through the through the arteries in the leg or in the arm, um, same as the bimetal stents. So self-expansion, as it is called, is required because of the procedure of minimally invasive insertion. So these are the requirements of the materials, and these are very, very challenging requirements. And many people started working on these biodegradable materials in 2000 and so did we. We developed a number of options. We developed the basic processes for cutting these stents, as it's called. Many of these stents have some very intricate patterns that need to be cut into the stent. These designs are generated by computer-aided design and then cut by a laser. So this is done for metal stents as well as for biodegradable stents. For metal stents, this process has been going on for a long time with carbon dioxide microsecond pulsed lasers, but we had to develop these procedures for biodegradable polymers. So if you try to cut stents made out of polymers with the same laser that is used for metal stents, you have a lot of collateral damage around the site of the cutting, so to speak. So microsecond lasers cannot be used for biodegradable polymers. We had to resort to what are called femtosecond pulsed lasers that cause minimal damage to the surrounding polymer after cutting these patterns. So this was not a trivial exercise. It involved a lot of trial and error. And we worked on it along with other people in the world to develop femtosecond laser cutting. We did a number of studies on the material properties. We characterized the memory effect, the self-expansion of these stents, control of the degradation, drug release studies in vitro. And here is Frank, who was involved with many of these studies in his PhD work. But the basic idea is that the development of the material takes a lot of preclinical evaluation. And so finally, the company we founded ended up with a material, PLA material, but with special attributes such as high toughness and strength, and the capability to be laser machined. It should have also programmable biodegradability over 12 months or thereabouts. So these are some pictures of these stents that are very flexible, can be employed or deployed on the surface of a balloon catheter, crimped and then self-expanded at the site of the blockage. The concept was licensed to a company called Amaranth Medical, as I mentioned before. It was funded partly in Singapore and partly in the US. It has undergone a lot of human trials all over the world now, not just in Colombia. That was the starting point. And many generations of these stents have been developed. But unfortunately, the story of biodegradable stents took a bad turn with the problems that were associated with the very first biodegradable stent called the Abbott Vascular PVS stent that was developed using a crystalline form of PLLA, polylactic acid. So they developed it by extruding the PLLA resin, oriented it, and then formed a highly crystalline tube. which was then cut according to the femtosecond laser process and then crimped onto the balloon. Amaranth, in contrast, developed what is called an ultra-high molecular weight PLLA polymer, which was not fully crystalline. In fact, it was highly amorphous that led to a very tough properties with very little breakage, as I will show later. But the scaffold cutting process and the crimping process were similar to the Abbott vascular stent. Okay, and this is important in understanding why the Abbott system failed and was withdrawn from commercial use after approval by the FDA. So it got approval in 2019. Sorry, in 2016, in fact, but was withdrawn in 2019. And I will tell you briefly why it was withdrawn. So this is the structure of the Abbott BVS stent made out of a highly crystalline PLLA scaffold. Okay, so the reasons that led to its withdrawal had to do with worse, longer-term outcomes, restenosis, thrombosis, revascularization as it's called, which is re-intervention in small diameter arteries compared to bare metal stents. It was also not endothelialized as much compared to, again, bare metal stents, which is a problem with clotting. So the thrombosis happens because of the lack of endothelialization. And endothelialization is also important for fully degradable stents, because if it is not endothelialized, then the degrading stent will actually put out pieces of the stent into the blood vessel, which is not at all desirable. It is a big problem if that happens. So endothelialization is key to the performance of fully degradable stents. And also what is called lack of anchoring to the vessel wall. So the material that expands with balloons also shows a little bit of recoil. So that means that it's not fully touching the vessel wall at all times and can migrate from the site at which it was deployed. And there were lots of problems with deployment and very different in deployability from metal stents. So these have all been covered in a nice paper in Nature Reviews in 2019. And those people also did some big studies to show problems with these stents. And one of the major problems was fracture. Fracture happens at the joints of the struts, which are stress concentration points. So in contact with bodily fluids, these stress concentration points fracture, and the number of fractures increases over time, as can be seen here. There are also other problems with deployment, such as overlaid struts and strut irregularities, which I will not talk about. But the basic problem is really the fracture, as well as the lack of endothelialization. The lack of endothelialization is caused by the thickness of the strut. So compared to metal struts, these cobalt chromium struts, for example, are about 80 to 90 microns. But the biodegradable stent strut is about 150 to 200 microns. Because it's made out of polymers that are lower in mechanical properties compared to metals, they have to be thicker to withstand the pressures inside the arteries. Okay, so the thicker strut also leads to disruptions of blood flow, as can be seen here, and creates stagnant regions of blood flow, which then lead to a series of activations of platelets that cause clotting and so on. Okay, so there's a coagulation cascade activation because of the presence of these platelets in the stagnant regions of flow. If you can make the struts thinner, the stagnant regions disappear, so you don't have this coagulation cascade, and there is no more delayed thrombosis, or there is actually a development of healthy endothelial cell coverage if the struts are much thinner in the region of 80 to 100 microns. So this is now well accepted and is led to many people working on lowered strut thicknesses for both the metal stents as well as for biodegradable biodegradable metal stents and biodegradable polymer stents. So you can also make biodegradable stents out of magnesium and iron, and many people are working on reducing the thickness of these magnesium and iron stents. The Amaranth second and third generation stents are much lower in thickness. In fact, the latest version is about 80 microns. Okay, and this is a very difficult thing to achieve with polymers. So this sort of shows the strut thickness reduction in the 3 generations of the Amaranth medical stents, all the way from 150 microns to less than 100 microns. Okay, and these have also undergone clinical trials, but unfortunately, because of the failure of the Abbott vascular stents and its withdrawal, the FDA is now demanding a lot more data, longer term data, before it can be approved. And many companies do not have the money or resources to continue these studies. Another way to address the problem of strut thickness is to develop nanocomposites. We are also doing this work now using nano fillers, which are also radio opaque. So I mentioned that all of these stents have to be radio opaque so that they can be seen in the X-ray. And we do this now by actually using barium sulfate or tantalum or other particles that will increase the mechanical strength while at the same time improving the radio opacity of these stents. Okay. So I'll just summarize the work here by showing the problems with the absorbable stents. So the radial strength has to be maintained for at least 3 to 6 months, and then they can slowly start to degrade, while the tissue coverage starts to happen at about the same time and must be fully developed by 12 to 18 months. Okay. In the meantime, the radial strength is decreasing. For fully degradable metal stents, it's decreasing faster than it is for polymers, which have this bulk degradation mechanism. So more difficult to control the mechanical properties for fully degradable metallic stents. But people are working on this, so there is hope that low strut thicknesses stents can be developed and tested in the clinic very shortly. Our work in biodegradable coronary stents also led to other stenting applications in ureteric stenting and tracheal stenting, where also things are blocked and need to be unblocked for a short period of time, and then the stent should disappear. But I will not go into those applications. But just suffice it to say that materials discovery and materials manipulation are required, because the degradation period for these different applications is very different from coronary stenting. I'll now turn to the second condition, which can be treated with biodegradable materials. And this is what we call the holes in the heart. So there are 3 different types of holes in the heart. Let me just have a sip of coffee here to keep me awake. Holes in the heart are defects in the heart that are either present at the time of birth or develop over time. So something that is present at birth is called PFO, or patent foramen ovale. This is simply a flap-like opening between the 2 atria in the heart that is always present at birth. So in the fetus, this hole allows for free exchange of blood between the 2 chambers, which is fine in the womb. But soon after birth, 1 month after birth, this should close, so that there is no mixing of oxygenated and deoxygenated blood. Okay, and when this does not close by itself after 1 month, then you have to go in and close it using interventional procedures. Similarly, there are other holes in the heart called atrial septal defects, which are larger in size than this one, which also need to be closed in patients. And there is also something called patent ductus arteriosus, which needs to be closed between the pulmonary artery and the aorta. Okay, so just 3 examples of holes in the heart, which is not very common compared to coronary artery blockage, but nevertheless a problem to be solved. And currently this is solved by using a nitinol mesh, as it's called, nitinol wire cage, which is covered with polyester fabric. And the reason for using the nitinol is that it can have self-expansion capabilities. And this is very important in this case because again, it is guided by the same procedure. So it is deployed through a hole through an artery in the leg or in the arm, and then guided all the way to the atria, and then put in place in the holes in the atria, and then released from the sheath. So it is kept inside a sheath until it reaches the site of the deployment. And only nitinol has these wonderful self-expansion properties among metal materials. The problem with the nitinol occluder is that, of course, it's permanent. Many people, nitinol is an alloy of nickel and titanium, and this causes allergic reactions to the nickel and causes clotting and blocks access to one side of the heart. That's the most important thing. So all of these interventional procedures these days involve the use of this artery in the leg, guiding to various chambers of the heart and addressing it through this procedure. So the presence of an occluder in one of the septa that divide the chambers prevents access to the other side of the heart. So if the occluder is in place, then the one side of the heart cannot be accessed by using this intervention. So if it can be made to be degradable, then you can have access to both sides of the heart, even if the occlusion happens. So the idea is to develop an occluder that is fully degradable in 6 to 9 months, can be deployed percutaneously. It's retrievable and repositionable, etc. It must be visible under X-ray and must be non-thrombogenic. So a number of, we have done a lot of work in this area and have published on the solutions to the problem. And this is sort of one of the devices that we have developed over time. It's a double umbrella design with one side having a film that occludes that hole, and the other side is used for anchoring or repositioning and so on. I will not go into the details of this, but this has been developed with fully degradable materials like polycaprolactone and polylactic acid. And this is sort of the picture of how it's deployed. So it's also put inside a sheath and then introduced percutaneously all the way to the site of the hole or the opening, and then deployed using a complicated procedure, which I will not talk about in detail. But once it's deployed, the sheath is withdrawn and the occluder remains in place and will slowly be covered with endothelial cells over time, and then it will no longer be needed. Then it can start to degrade. We have done animal studies to show the occlusion, successful occlusion at 1 month. And this is ready to be translated, but because of the problem of the market size, as I will explain later, there are very few people interested in funding this sort of effort, which is unfortunate, but because this is a much needed solution right now for many, many patients. The third device that I want to talk about is Embolic Plug, as it's called, for endovascular embolization. So this is a device that actually induces a clot at some location where it's desired. And in aneurysms, for example, so many people develop aneurysms, which is the development of a sort of a protrusion in unhealthy blood vessel, which due to high blood pressure expands and becomes sort of a balloon here. And then eventually it'll burst, giving rise to strokes. And when this happens in the brain, this is very, very dangerous. So one way of stopping this from growing is to put in a plug here, embolic plug as it's called, that prevents access of the blood to this ballooned area. So if detected early enough, this can be done. The plug preferably prevents this access, like I said, and but does not prevent blood flow through the core of the blockage. But there are also other cases where a plug is required, but only temporarily. And I will talk about this. And this is for cancer treatment, as it turns out. So particularly for liver cancer, what you need, because anything that is administered to the liver usually is cleared by the liver very quickly through clearance by the hepatic artery. So in order for the drugs that are administered to the liver or the radioactive particles that are administered to the liver, in order for them to have some duration of action, you have to block this clearance by the hepatic artery, at least temporarily, while this procedure is happening. So blockage of the hepatic artery for these two treatments, selective internal radiation therapy, SIRT, or transarterial chemoembolization, is required to enhance the efficacy and duration of action of these procedures. Okay, either radiation therapy or chemotherapy. So you need something to temporarily block the hepatic artery, and then you can restore it once this degrades, so that the procedure can be repeated again and again until the liver cancer is sort of kept under control. Right now, there are embolic plugs available through the use of metal coils, but these are permanent. Gelatin sponges, but unfortunately, this is uncontrollable target occlusion, not predictable, very variable. And the degradation is also quite variable because of the presence, because this is enzymatically degraded. The collagen or gelatin is enzymatically degraded, so you cannot predict how long it'll take for them to degrade. So we wanted to develop a temporary self-expanding plug with a predictable biodegradable profile, which is also radio opaque and can be deployed in such a fashion to completely occlude the target vessel. Okay, I won't go into all the details of what we develop, but it involves the use of shape memory polymer. So in this case, the polymer itself is made out of PLGA and can be made to have memory. So basically, the polymer itself is extruded as a monofilament, and then it is subjected to some procedures which allow it to have shape memory, which is the uncoiled configuration. Okay, and then it's coated with a hydrogel material, polyethylene glycol diacrylate, which is cross-linked, that can swell after introduction into the blood vessel. Okay, so the material is inserted in the catheter as a rod of PLGA surrounded by this hydrogel, which is dried. Okay, so it's a very thin rod, monofilament, that is introduced to the blood vessel. Once it touches water, it allows for expansion. As at the same time, the biodegradable polymer core undergoes what we call buckling, and that gives us mechanical strength. It anchors this plug in place, which is, the plugging itself is done by the hydrogel that swells and locks the entire segment, while the polymer is present as a coil to give it mechanical integrity for as long as the polymer is not degrading. Okay, so these are the polymers that are used in this application. Poly lactic co-glycolide, PLG, a copolymer that can be filled with radio-opaque fillers like barium sulfate and so on. And then the polyethylene glycol diacrylate, which is the hydrogel that swells to plug the artery. Okay. I won't go into all the details of the evaluations we did to evaluate the radio opacity under X-ray, as well as the mechanical properties and self-expansion or shape memory properties. But this is the composition we ended up with. Bismuth oxychloride nanofiller inside a PLGA system, and the filler content is about 50%. And we did a number of shape memory studies and recovery studies, which I will not go into, and then developed in vitro evaluation of the time it takes to completely block the vessel. And then we went on to an in vivo study inside a porcine pig model. And I will show briefly what happens in this, in a rabbit model. First, the blood vessel to be embolized is located by injecting a contrast solution, as you can see here. And then the coil is, the device is introduced as a rod, and then it starts to coil. And then the vessel is slowly occluded within about 30 or so seconds. Reduction in flow starts to happen fairly quickly, and eventually it's completely blocked, as you can see. Okay, as I hope you can see. So that sort of is all I wanted to cover today. And this is a summary slide that talks about the evolution of biomaterials that are a key part of all of these devices, okay? And in general, in cardiovascular implants, there is a move from biostable to biodegradable materials. And that requires a lot, historically, most of these materials innovations came from companies, such as, for example, in the development of the balloon catheter and the first stents and occluders. Many academicians or academic research started around 2000 when the need for biodegradability was first mooted with the work of Igaki Tamai in Japan and our own group on stents and occluders. And then now the range of fully degradable implantable materials is still very limited to the PLA type materials. We need newer materials that can have shape memory properties and so on. And one of the lessons we learned is that in order for academic research to be translated into products, most of the investors are only interested in big market products such as coronary stents and so on, not even the ureteric and other stents, which are very small. So there is a difficulty in attracting funding for the smaller market-sized devices such as occluders. We struggled for quite a long time to get funding for this. Unfortunately, we're still unable to get some funding for this area. We are going to keep on trying with something like the Bill Gates Foundation or other charitable foundations that might be able to help us out. But so far, no luck. I want to first acknowledge the entire team, finally acknowledge the team that worked on various aspects of this. I have not put in Frank's picture here, unfortunately, but he also contributed to many of these areas of research. This is the occluder team. We had a lot of collaboration with clinician scientists. That's one of the ways we work. With Dr. James Yip from the University Heart Center, Philip Wong, National Heart Center, and then a cardiologist at the Apollo Hospital in India. I'll end up with asking you if you have any comments or questions and stop the sharing of the screen here.
Thanks a lot, Shugo.
Thank you.
You didn't put the picture because maybe, with my hair, they may not have recognized me from that time.
That is true.
So yeah, I hope we have some questions from professors, from students. Our school is, working on a master program on biomimetic materials, biomaterials.
Mm, mm,
So hopefully this will come out at some point.
excellent. Yeah. Yeah. So we are also doing a little bit of work in that area. Maybe another seminar on this at some point.
We have a question in the chat.
Yes. So the question reads, besides degradable PLLA polymer and the nanoparticles that have been used, which other polymer have you considered for stents, and how do you see the future of these hybrid nanomaterials? That is a very interesting question. So uh, for biodegradable stenting, most people have concentrated fully on PLLA type materials. And these are not just the PLLA polymers, but also copolymers of PLLA. There have been people working on metallic stents that are fully degradable, and that is either magnesium or zinc, which is now becoming very popular, or iron. So all of those have been considered for stents. But in the case of the metallic stents, the degradation has to be controlled by the use of polymer coatings. Without that, the degradation profile is not acceptable. So that continues to be an area of interest. And in particular, zinc is going to be of great interest in the future. Other polymers that have been considered include chitosan, which is one of, I suppose you could call it a biomimetic material because it's naturally produced. But still, there is nothing like the PLLA and PLGA family that have controlled and predictable biodegradability. So these degrade by simple hydrolysis and bulk hydrolysis. So bulk hydrolysis is also important. If you recall what I showed on the biodegradation profile, it is best if the mechanical properties are stable for a long period of time and then for them to collapse rather than this linear degradation profile that metallic stents exhibit. So this is the problem. So can you develop bulk degradable materials such as the PLLA family that can also have good mechanical properties over time? That is a challenge. I hope there are some chemists among you that can work on this sort of problem. Okay.
There is another question, actually from Antonio again, about the intellectual property.
Yes, Antonio, yes. Okay, so this question is about how do you handle intellectual property at the university? What are your learning and goals reached in your university and your recommendation in establishing this type of committee? This is also a very good question. So translation of academic research from my own experience at NTU and now at NUS, and knowing a little bit about how it is done in the US universities, I think this is going to become very, very important in the future. Many of the companies these days, especially the big device companies, have stopped doing their own R&D. Instead, they are looking for ideas generated by either smaller companies or by academic researchers. So that's why this is going to become more and more important. But what the committee or the intellectual property committees or technology translation people in universities should do is to fund the research up to a certain technology readiness level. This is called TRL. And TRL levels of about 7 to 8 will attract attention by the big companies or by investors to develop the technology further for clinical trials. So this is all preclinical research, which must be funded some way by National Research Foundation grants or NIH grants or similar organizations within the countries, different countries that exist, but also by the universities themselves. They can also fund some of these research that are promising. So that is what we are encouraging in NUS, is to develop or find funding to develop these ideas up to a certain technology readiness level, then license it to companies or to VC firms. This is how we handle the technology property in the universities. We have technology managers that are assigned to each invention disclosure that is submitted to the university, and they guide the researchers to patent and to also explore applications that they might not have considered when they first filed the invention disclosures. And then they also help with the licensing and also finding funding to develop it to different levels of technology readiness. I hope that answered your question. Another question here is, do any of the materials you work with attract microbes and formation of biofilms? This is also a very important question. In a sense, the drug-eluting stents will probably not attract many biofilms because they have drugs that are either cytotoxic or cytostatic. Unlikely for them to attract any kind of biofilms in situ. Medical devices that in general have been implanted will attract biofilms. And of course, the biofilm formation is started by sort of unhygienic procedures adopted by the cardiologist or the interventional scientist, or sorry, the interventional clinician. So if that happens, then of course there is no option except to either treat systemically with antibiotics, or actually removing the device, unfortunately. So one way to prevent this from happening in situ is to develop these coatings of cationic polymers, which can also be the carrier for the drugs and so on, if drugs are required, or in the case of bare metallic stents, use cationic polymer coatings. That's what many people are trying to do now. There is another question from Frank, which asks about radio-opaque polyesters without fillers, and it allows to image polyesters in deep tissue and see cracks using X-ray, fully degradable. Might be interesting to discuss more. Yes, I agree. So one early example of this sort of radio-opaque, actually polycarbonates, developed by Joachim Kohn at Rutgers, it has been adopted by one of the companies. I forget the name of the company that is developing this further. But the idea of using it to image in deep tissue is very, very interesting. And if you can follow the degradation over time in a non-invasive way with imaging, that'll be very, very exciting indeed.
Yes,
absolutely
actually,
right.
We show that in rats. We can measure, we compare the in vivo and in vitro degradation using X-ray and non-invasively, and we showed it.
Yeah. So if you can also image it using optical coherence tomography, that'll be really very important.
Mm-hmm.
And it'll also help the approval process, to be quite honest. if you can show that degradation happens only after full endothelialization, then it will be easier to convince the regulatory agencies. Okay, sounds like that's all. Another question from Frank, which is, do you see opportunities about biodegradable natural polymers from plants? You mean like cellulose and other types of materials?
Yeah, that's right.
Okay, yeah. So I don't know if you know about this nanocellulose that has been developed by a few researchers, but as fillers, these can be very interesting to enhance mechanical properties. Fully degradable stent, for example, made out of biodegradable natural polymers could also be interesting, provided the mechanical properties are met. So chitosan, which is animal derived, has very excellent mechanical properties because it forms the crab shell material, which is hard and yeah. And if cellulosic materials can attain that level of hardness, it can be very interesting indeed.
Yeah, because we found that depending on the source of the cellulose, we have different degradation time, different degradation kinetics in the same conditions. So, so maybe
something new to be explored. Yes.
Maybe in the future. Well,
yep.
You had a lot of questions. Thank you so much, Subbu, again, to take this time early in the morning.
Thank you.
Late at night for us, for our professors and students. I hope we can continue this discussion online and offline. And we are
online, offline. Maybe a conference in the Galapagos Islands at some time.
That's actually, you have the organizer in this chat, so I think he's here.
Okay.
I hope to see you. Bye-bye. Thank you so much.
Thank you. Take care, everybody. Stay safe.
Thank you very much.